ORDA Science Webinar. Edge topic, Microbial Heroes. Working with microbes to solve and respond to anthropogenic impacts. July 13, 2022, 18:05 Coordinated Universal Time. Recorded and organized by Samantha L. Forster in Microsoft Teams.
And thank you, Samantha, for starting that recording.
Title slide with logo USGS Science for a Changing World. ORDA Science Webinar Series. Dr. Denise M. Akob, U.S. Geological Survey. Denise's headshot is of a dark, straight, short-haired, smiling woman with a blue blouse in front of a flowering bush. The findings and conclusions in the webinar are those of the presenters and do not necessarily represent the views of DOI's NERDAR program, the department, including the trustee bureaus, or the United States. Although some of the presenters may be employees of government agencies, the ideas described herein do not necessarily reflect the policies of those agencies, and no official endorsement should be inferred on NERDAR.
And it will be posted at a later time, and we'll try to get the slides up as well. So I had the opportunity to talk with Denise last week, maybe, I guess it was already, and she has a real passion for microbes, so I'm excited to hear her talk today and teach us a bit more about what microbes may mean for NERDA when it comes to what they could tell us about anthropogenic impacts in the environment. And with that, I will go ahead and introduce Denise. Dr. Denise Akob is a microbial ecologist and biogeochemist whose research aims to understand interactions between microbes and their environments in order to solve anthropogenic issues. She obtained her PhD from Florida State University in 2008, then was a postdoctoral research fellow at the Friedrich Schiller University in Germany prior to joining the USGS in 2012. Denise has published over 60 papers on uranium bioremediation, microbe-metal interactions, subsurface biodiversity, and environmental effects of oil and gas development. She received a USGS Early Career Leadership Award in 2017 for her outstanding service and scientific achievements in representing the agency. She also co-leads the USGS Environmental Health Energy Integrated Science Team Project, which aims to understand the environmental health impacts associated with the life cycle of energy resource development. And for those of you who join this series regularly, you know I like to have a little fun fact about the presenter, and I really like Denise's. She has 2 science tattoos, and one of them is highlighted in the book Science Inc. by Carl Zimmer, which is a collection of science tattoos from around the world. So after I read that, when Denise sent me her bio, I looked that book up. It looks very interesting. So with that, Denise, I've stopped sharing. I will hand it over to you. I'll go off camera. I'll do a quick mic check for you, and then you can just get started.
All right, great. And I'll go ahead and start sharing. And thank you again for the introduction, Joelle.
2 speakers are displayed. Jo Ellen is a woman with long reddish medium brown hair, wearing glasses and a striped blouse, sitting in front of a paneled landscape picture. Denise has a blurred background with her dark hair pulled back, wearing a pink shirt.
Oh, sorry about that. There we go.
Title slide is displayed. Edge topic, Microbial Heroes. Working with microbes to solve and respond to anthropogenic impacts by Dr. Denise M. Akob, she, her, hers, U.S. Geological Survey, Geology, Energy, and Mineral Science Center, Reston, Virginia, U.S. Department of the Interior, U.S. Geological Survey, USGS Science for a Changing World logo.
It's always a little clunky to share from a single screen. There we go. Can you hear and see me okay?
Looks great.
All right. Well, thank you again for having me. I'm really excited to be able to talk to you today about what I like to think of as microbial heroes. Hopefully, everybody was pretty interested to find out what microbial heroes are, and that's what brought you to my talk today. So my expertise, as Joanne said, is in microbial ecology and biogeochemistry. So what that means is that my work really sits at the interface between microbiology, hydrogeology, and geochemistry. And so my work is always aiming to understand how microorganisms are affecting their environment and vice versa, how the environment is affecting microorganisms. And we want to do this because microbes can be little heroes and they can catalyze a lot of reactions that will affect human-made contamination and also they can affect environmental health and water quality. And specifically, microbes can do this by my really pretty purple microbe here. And that is, it's very similar to one of my science tattoos, actually, I should mention that. But the way that microorganisms can affect the biogeochemistry and the environmental health of where they live is by eating different food sources and breathing different electron acceptors. So I always explain this to my kid and her friends that, we like to eat macaroni and cheese and pizza, which is this generic CH2O molecule or carbohydrates. And so microbes always need to eat something. That's how they sustain life. But we also need to breathe. We breathe oxygen. But a lot of the microbes that I study will actually breathe heavy metals, uranium, other radionuclides, and other types of compounds that can be contaminants that will threaten human health and water quality. And so what I'm going to talk to you today is about how different microorganisms can eat different contaminants in the environment. But if you're interested in hearing a little bit more about other work that I've done, I can talk about how organisms are actually breathing different contaminants. So just follow up with me. And so this is kind of the basic, building blocks of everything that's living. And we can exploit these naturally occurring processes to actually improve environmental health and affect water quality.
Microbe eat to gain energy where CH2O turns to CO2. Breathe. Respiration goes from O2 to H2O.
So why microbes? Well, yes, this is a giant pile of stuffed microbes. I have this giant pile in my lab, and it's a great tool for actually doing outreach and connecting with young students and trying to get them excited with science. And so our giant pile of microbes, they're found everywhere. So this diagram by Caviccioli et al. 2019 shows all of the different habitats, which is every single habitat in nature where microbes can live. So that includes aquatic organisms, tourisms. There's even microbes that are being found to live in the air. And by those life-sustaining processes we mentioned earlier, these microbes can affect how different types of organic matter is decomposed, how much nitrogen or phosphorus is available to plants. They can actually form the base of the food chain, not only in aquatic systems, but even in soils. And they have a really important role in semi-arid and arid regions and habitats. Many of you might know that in the wastewater treatment plants, where all of our waste as humans go, microbes are our little heroes. They're the ones that are breaking down a lot of those waste products and allowing the water to be purified and move back into the water cycle. So when we think about remediation and restoration, we can harness the power of microbes being everywhere and being naturally occurring and their naturally occurring reactions to help in remediating different contaminated sites. So throughout my career, I've been looking at how microbes can basically catalyze reactions of taking carcinogenic or toxic compounds to something non-toxic in the environment, and how that can then fit into an overall restoration trajectory. So this is actually a site that I worked in Germany, where it was a former uranium mine, and it was actually restored back to a grassland. And we were looking at what types of microbial processes were helping to promote the success of those activities. But in order to have successful remediation and restoration strategies, we need basic science. We need to know a lot about microbial metabolism and how that metabolism actually interacts with the environment. So we know that microorganisms are everywhere, but we don't know the full diversity of all microbes, and we definitely don't know all the different reactions they can catalyze. So my team alone has discovered some pretty novel metabolisms, and we're learning how those really interesting metabolisms can have important effects on environmental processes. So when we do my environmental microbiology, so going all the way back to the beginning of the slides, I called myself a microbial ecologist or a biogeochemist. You can also say that I'm an environmental microbiologist. So a lot of these terms are interchangeable, but they have a little bit different meanings for different folks in their specialties. So And we're thinking overall about trying to understand microbes in the environment. We're thinking about microbial populations, so not just a single organism, but lots and lots of organisms in a single sample. And when I say lots and lots, like we're talking about thousands of species can be in a single soil sample. And then there can be millions to billions of cells within a single soil sample. So what we want to know is in that environmental microbiology framework, who's there? So who are the microbes? What's their identity? How diverse are they? So how many different types do we have and who are they? We can do this by looking at microbial biomarkers. And so what my team primarily looks at is the DNA of the microorganism. And we specifically look at something called the 16S rRNA gene. This gene is highly conserved, meaning that if that gene is not present, so if it would evolve to disappear from the genome, the microbe can't live. And so it contains a lot of information about that can be utilized to figure out who's there. We also want to know what are they doing? So just because you know microbes are present in an environment, that's only one piece of the puzzle, but we really want to know what can they do? How can we utilize these organisms to solve problems? How are they contributing to the restoration trajectory or natural processes in a pristine environment? So we want to know their function and activity. And we do this by using an experimentally derived approach. So a lot of the work I do brings field samples back to the lab. We run experiments where we look at how geochemistry changes as microbes are breathing and eating. And we'll often even culture microorganisms to try to get down to a single organism that we can then understand more about their genome or their very specific physiology. In a lot of cases, though, some of the experiments I'm actually designing for a field work trip in August, we're really trying to look at what's the activity at a whole, like on a whole ecosystem level for the microbes, because in situ, in an environment, they're all going to be working together to have a function and activity. And so a single organism might not be really representative of what's happening at the site.
Microbe cartoon with an oil drum, a copper spool, a lead brick, and liquid mercury. 3 microbes are wearing capes and holding up the earth.
So what I wanted to talk to you all about today was work that I've done on microbes and spills. And I found this really awesome graphic, thinking about microbes and water quality and how they're interacting with things like oil spills or heavy metal contamination in the environment. And then, of course, we have our little microbial heroes. And honestly, I could not have drawn this little graphic better. So I was really pleased that I could borrow it from this artist. So I'm going to talk to you today about 2 case studies on microbes and spills. They're all related to U.S. Energy production within the United States. So as all of us know, energy resources are still dominated by fossil fuels as we're on the trajectory to be utilizing primarily sustainable energy resources. And, this graph from the U.S. energy information industry shows the percent of U.S. energy production by source over time and going back to the 1950s to about 2010, 2015. And we see that, renewables or sustainable energy resources are a much smaller overall percentage compared to things like natural gas, coal, and crude oil. So these fossil fuel resources are still a really important part of our domestic energy production, and understanding what types of environmental effects can come from those is still really important, again, as we're on that trajectory. So one of the things to understand is that natural gas, coal, and crude oil is very chemical and water intensive. And so there's a lot of potential for spills of different fluids from these production processes. So I'm going to talk today first about a coal processing chemical spill that was into the Elk River, West Virginia. This was actually a NERDAR case that was recently closed. And then I'm also going to talk about some current work that we're doing, trying to understand how wastewaters from dumping of oil and gas wastewaters from crude oil and natural gas production, what's happening when they're illegally dumped on the landscape. And while this is not like a neat chemical spill, it is a complex water source that has both naturally occurring from the formation where oil and gas is being produced and chemicals used in oil and gas production. And so I thought that would be a really important vignette to talk about for examples of how our science is important for understanding spill effects. So I'm going to first talk about the spill of this chemical MCHM. The full name of the compound is crude 4-methyl cyclohexane methadol, definitely a mouthful. The crude MCHM compound, and I will refer to it as MCHM, MCHM from here on out. This crude mixture, is a mixture of several compounds, including cis and trans isomers of the 4-methylcyclohexane methanol. And this compound is used as a frother during coal washing to remove impurities. As you might know, when coal is being mined, it goes through a processing step to remove impurities that could become environmental hazards during coal burning. And so things like silica and pyrite and clay are removed through a frothing agent, also known as MCHM, and that's demonstrated in this upper right-hand panel. So that's one of these natural or very standard processes for the coal. The compound, though, that we're talking about, because we're focusing on spills today, was stored along chemical storage tanks, as you can see in this bottom right photo, right along the Elk River in Charleston, West Virginia. This is a site that was managed by Freedom Industries. And as you can see, it's very, very close to the actual river right on the bank. In January of 2014, one of those storage tanks had a catastrophic failure, and about 10,000 gallons of the crude MCHM compound spilled from that tank and went down the river embankment into the Elk River, West Virginia, which then resulted in the contamination of drinking water supplies for over 300,000 residents within that area. At the time of the spill, there was little to no information available on the toxicity or the fate and transport of the compound. So this was a pretty major environmental disaster that ended up having a murder case and having a lot of public concern, public interest in what was happening and what the risks were to human health, but also the habitat. So what we wanted to know, USGS did some sampling and developed some methods for looking at MCHM compounds and being able to detect it in the environment. But the team that I collaborated with, we were really interested in knowing what is the long-term fate of the compound in the environment. Is it sticking around for a long time? Is it degrading very quickly? Those kinds of things. And so this diagram here on the left, so I'll start on the right-hand side, is just an image of the compound. There's the trans isomer at the top and the cis isomer at the bottom. And on the left here, I'm showing all the different processes that are proposed for different chemicals in the environment. So if a chemical is spilling into a water body, it could dissolve and just spread downstream within that water body, but it could also evaporate or be oxidized due to sunlight. However, the compound could also be biodegraded within the water column. It can also just disperse within the water column, and it could also absorb to sediments and even undergo biodegradation there. However, with this compound, like I said before, no one knew what would happen to it. Is it moving down gradient and now being a threat to downstream drinking water supplies? Or is it something that's being attenuated very rapidly in the environment through some of the other processes? So we sought out to look both at adsorption to the sediments, and I'm not going to talk about those data today, but I'll show you a screenshot from the manuscript where we published these results. I'm going to focus today on the work where we were looking at biodegradation. And so we were looking actually at biodegradation in the sediments. So sediments generally serve as a long-term integrator of the environment. So if we think about a water body, any microbes or chemicals that are present in water are going to be moving with just normal water flow. But microbes that sit in the sediment are going to be integrating a signal because they're going to be exposed for a lot longer term, and they're going to see compounds just kind of interact and sit there a little bit longer. So we primarily looked at biodegradation. So we looked at biodegradation in the sediments of the Elk River. And we were interested in thinking about how our microbial hero could actually eat MCHM. So there they would be producing CO2 as a byproduct of eating. And then how they could breathe iron, so breathing iron oxide, which is rust, and how they can convert that to reduced iron 2. So these are organisms that do not, their metabolism is not favored under oxygen conditions or oxic conditions. They really prefer to breathe minerals that are in the sediment. So our work was published in Environmental Science and Technology in 2017. Isabel Cazarelli and my collaborators I'll mention here, and Isabel was the lead author on this paper, and this is work that was done through both of our teams and the interdisciplinary combination of doing biogeochemistry with the microbial activity. It was a really robust way of knowing what would happen to this compound in the sediments.
Tidal degradation of crude 4-MCHM in sediments from Elk River, West Virginia by Isabel Cozzarelli, Denise Akob, Mary Jo Baedeker, Tracy Spencer, Jean Gishk, Darren Dunlap, Adam Mumford, Amisha Porritt-Peterson, and Douglas Chambers.
So for the biodegradation experiments, again, how are the microbes eating this compound? We went out to the field. Here is a Google Earth image of the Elk River. Elk River is flowing from the top of the picture down to the left. So this is the north here. The white tanks on the riverbank right here are the Freedom Industries site. And I believe the tank, like right kind of in the upper middle, is the one that had leaked. Oh, I'm sorry. It's up here on the furthest upstream tank is the one that had leaked. My mistake. So we went out to the field and we collected river sediments from a background site, which was upstream from the Freedom Industries location. So these are river sediments that were not affected and not exposed to the compound. And then we also collected samples right within the boom that had been put there for immediate response to actually try to prevent any of the compound that was floating on the river surface from moving downstream. And so right where the spill had actually directly hit the river and the sediments. So we collected submerged sediments from there. We brought these sediments back to the lab. We incubated them in these microcosm bottles. So there's actually clean river water from the background site and impacted or background sediments mixed. We had a number of treatments. All of our treatments were stored anoxically or incubated anoxically, so there was no oxygen present. We had treatments with MCHM added. We had unamended samples, so where we just had the sediments with no compound added. And then we also did killed controls, and that's important for us to be able to assess whether or not a process that we're observing is due to the activity of the microbes or to other chemical reactions just interacting with the sediments or something like that. So we did this. We had both background and impacted treatments, impacted samples times each of these treatments, and everything was incubated in triplicate. We then measured geochemistry over time. So we wanted to look at the iron that the organisms were breathing. We also looked to see whether or not they were breathing sulfate and how that MCHM compound was being degraded. We also did microbial community analysis. So this is the who's there part of the study, while at the top, that is the what are they doing. So microbial community analysis, I'll go into a little bit more detail, the methods on our next case study. Well, we use this magic box or this magic method in a way called Illumina 16S I-tag sequencing, where we sequenced all of the specific gene of the entire microbial community. We do a lot of bioinformatics with my little silly cat gif here, and we can then look at what is the composition of the microbial community, who's there, how diverse is it, and what are the different taxa.
6 clear bottles with our liquid and sediment. The first 3 labeled background, and the last 3 labeled impacted. 1 bottle from each the background and the impacted group are labeled by the star.
So this is an image of our microcosms. These are 6 microcosms. The first 3 on the left are microcosms constructed with sediment from our background site. And then the ones on the far right, 3 are from our impacted site. All 6 of these bottles were actually had MCHM added to them so that we could really look at the process of biodegradation. You'll notice that 2 of the bottles actually have a slightly different color. So because MCHM, the specific properties of this organic compound, we had to design a special bottle system that we could sample in a way that the microcosm material would not come into contact with like rubber stoppers. So we had these special sampling ports that we DIY'd up at the top, and they did a really good job for the majority of our bottles, except for a few, at keeping the conditions anoxic. So we were therefore promoting microbial activity that was not coupled to breathing oxygen. And we see in the bottles where we were focusing on those anoxic anaerobic metabolisms, that we see this nice dark brown gray color on those bottles. And that's indicative that the microorganisms were breathing iron and creating a new mineral which had more of like a black brown color to it. However, we actually had leaking in some of our bottles, so the ones that are shown here with the yellow star, and you notice that they're a little bit more orangey in color. And so that was our first observation that we probably actually had degradation coupled to oxygenic activity, so aerobic activity. And we were able to do some different measurements on the gas headspace and find that that was true, that we had oxygen leaking into these bottles. Well, it ended up being a really great accident that that happened. So on this plot, I'm showing time of incubation on days on the x-axis. And on the y-axis, I'm showing the concentration of cis-MCHM. I'm only showing one of the isomers. We were measuring both, and the overall patterns are identical with the two isomers. So I'm just showing one plot for simplicity. And normally when we would do these types of experiments, if we had triplicate bottles for a specific treatment, we would actually average them. But what happened is that the two bottles that had oxygen leak in and we had aerobic activity, we actually saw that degradation went much faster. So if you look at the blue circle and the purple triangle, those are the bottles that I'm showing right here, where we had degradation of MCHM very fast. So within 4 days, all of it was below detection. So when we published these data, we actually published these as individual data sets because the individual observations were really valuable for understanding the dynamics and the function in this area. When we looked at the site-specific activity, when we look at our impacted bottles, so this is sediment from that impacted site, the microorganisms were previously exposed to MCHM, we saw that degradation happened second most also happened really quickly. So we had, in the presence of oxygen, we had degradation in less than 5 days, and then about 8 days for our previously exposed sediment microbial communities. When we looked at our background sediment bottles, we actually had degradation within 13 days. So what does this mean to us? Well, it means that our biodegradation, again, is fastest under aerobic conditions, but anaerobic degradation can still be occurring in these impacted sediments and is being coupled to iron and sulfate reduction. The really interesting thing, though, is that prior exposure to MCHM allowed the microorganisms to do this very rapidly. So it indicates that there were, in the river sediment, there are microbes who are very happy to eat this compound. And prior exposure to the compound will actually drive those reactions to occur more rapidly than naive microbial communities. So then we wanted to know, well, who are the microbial communities? So we now know they're functioning to biodegrade this compound of concern, but now who are those taxa? So I'm showing you here, this is a relative abundance plot. We're looking at the percent of total sequence reads for each microbial community, and each of the different bars and colors are a different taxonomic group. I know this is a very complicated plot. It's not one of my favorites, but it's one of the best ways we can show our overall snapshot of the microbial community. And so I'm providing it really as just a demonstration of the type of data we're generating. I'm going to walk you through a few more things, and then I'm just going to highlight some of the main findings from this work. I have here in brackets showing that the bottom data series are from our microcosms that we incubated without additional MCHM, and then our microcosms where we had MCHM added, and then our sediments, in situ sediments we collected at the time of sampling to find out what is the in situ microbial community look like. The impacted versus background bottles are indicated by the red squares and blue circles respectively. But the main thing that you should really take away is you're probably looking at this and saying, Denise, these don't look very different. There's a whole lot of orange, and then I see a whole lot of all these other things. we're not seeing giant changes. And that was really surprising to us. We expected our impacted in situ sediments to be very different from what we're seeing in the background. But the thing to understand is that this is done at the phylum level. We're at a really high taxonomic level. And so what I'm going to show you on our next slide is at this really high taxonomic level, it's really hard to see differences in the microbial community. What we are finding is that the proteobacteria is the most abundant group of microorganisms present, but this is a giant group of microbes. They're one of the most abundant groups of microorganisms in most environments. We're also seeing that the majority of organisms are related within the domain bacteria.
Bacteria dominated the microbial communities, less than 2% archaea, and taxonomic composition was similar at the phylum level.
There was many fewer within the domain archaea. So we did another analysis. So here I'm showing you a plot to look at how the overall microbial community might shift based on exposure. So this is using the same data set shown on the prior plot, but it takes into more consideration what the similarity between the different samples are and how the relative abundance, but also the taxonomic diversity, kind of putting that into a matrix, and we're looking at it from a multivariate in a multivariate space. So this plot is a non-metric multidimensional scaling ordination, or an NMDS. And each of these individual dots, and again, this is a demonstration graph, but each of these dots are a single microbial community. So an entire one of those microcosm bottles, we extracted DNA and we sequenced and got this, did this similarity analysis to see how similar that community is to another. And if you remember before, we saw very high taxonomic level differences. But when we look more at kind of the, look at all the different taxonomic levels, which is being shown, goes into this analysis, we see differences in the community structure based on the in situ exposure. So what I mean by that is everything on the left that's kind of highlighted in blue, except for one outlier, that always happens with microbes, they don't all behave. Everything highlighted in blue are background sediments. The ones in the dotted line were background sediments that were taken at the time of sampling, whereas everything in the solid blue circle are the microcosms constructed background sediments. On the right-hand side of the plot, we see our impacted sediments. And the first thing to look at is our in situ communities that are here in these dashed line and dashed line started out very differently. So in taking into account genus and species level differences, we're actually seeing shifts in the microbial community structure that just aren't evident purely looking at taxonomy. And what's also interesting is that, the background microcosms are changing relative to the background sediment, and that's likely due to exposure to MCHM in some of those bottles, but also to the bottle effect of incubating them in the lab. So taking all these different data together, we found that activity is much higher and faster in our impacted sediments, and that our impacted sediments have significantly different microbial community structure compared to our background sediments. So that indicates that exposure is shifting the microbial communities and also priming them for MCHM degradation. And so we're seeing the differences in these community effect is really providing evidence for an ecosystem level effect. So if you think about microbes in the sediments of the river as catalyzing really important ecological processes, this can be kind of your first level, lowest food web level effect on the community due to a spill and due to a contamination event.
In conclusion, MCHM rapidly biodegrades under both oxic and anoxic conditions. The spill likely attenuated rapidly in Elk River surface sediments and river water. Microbial communities in impacted sediments are primed for MCHM degradation, possibly due to selection from the spill. And despite the potential for rapid degradation, sorption of MCHM to sediments poses a risk for continuing contamination.
So to summarize that, this study, like I said, we were out looking at the spill of our crude for MCHM. As I mentioned, we did look at sorption. We found that the compound can sorb to sediments and potentially even be a long-term source of MCHM to the environment. if conditions would change and allow the compound to release. But we also found that biodegradation can occur very rapidly. That can happen under both oxic and anoxic conditions. Sediments tend to be anoxic depending on the local environments and water flow. And because biodegradation occurred so rapidly, it's likely that the spill attenuated really quickly, that the compound was not sticking around for very long term, that probably within 2 weeks, anything that was still around on the sediments, the majority of that contaminant mass was degraded. And just to reiterate this one point, that the communities that have already been exposed, they're primed, they're ready to go if they would have a second exposure event where they can degrade this compound. And so the selective pressure of a spill is going to be selecting for microbial communities that are well adapted to deal with the contaminants. So now I wanted to move on to talking about our more current work looking at illegal dumping of oil and gas wastewaters. So oil and gas development is occurring across the United States in many shale gas plays and other energy resources across the United States. And we've been working primarily in the Permian Basin, which underlies parts of New Mexico and Texas, and it's the single most productive tight oil province in the United States. It also underlies part of the Chihuahuan Desert ecoregion. So this is a really important part of habitat within the United States and Mexico. Within the United States, within North America, it's the largest desert. It's semi-arid and has some of the highest biodiversity of any desert that has been studied. And as this unique semi-arid habitat, it provides key ecosystem services as well as cultural and commercial services. So illegal oil and gas wastewater dumping. So our colleagues at the Bureau of Land Management started noticing in around November of 2017 a rash of illegal oil and gas wastewater dumps. So oil and gas wastewaters are primarily transported, at least at that time, using trucks, although they have also implemented in that region quite a lot of wastewater movement through pipelines now. And these wastewaters are characterized by having high total dissolved solids and variable concentrations of hydrocarbons. So in the transportation process from a well where these oil and gas wastewaters are being generated, the trucks, instead of driving them all the way to a wastewater disposal location, they are pulling off to the side of the road and releasing these fluids onto the desert landscape. So that's what all of this very dark brown desert is showing you, that this is a region that was affected, or an area, that was affected by that dump. By the time that we sampled in March of 2018, BLM was aware of 39 sites within a 5-mile radius, and all indication is that dumping is ongoing even now 4 years later.
Preliminary information subject to revision, not for citation or distribution.
So as I mentioned, oil and gas wastewaters, these can also be called produced waters. These are a combination of the hydraulic fracturing fluids that are injected into a well to stimulate production, and also water or brine from the formation. So hydraulic fracturing fluids can contain water, proppant, like sand, and a number of chemicals that will enhance production. In the Marcellus Shale, I do apologize, I did not update this for this region. Marcellus Shales in Pennsylvania, an average well is producing about 1.3 million gallons of wastewater over its lifetime. So these wastewaters have complex chemistry, and a lot of fluids can be produced that require handling and disposal. The wastewaters can have low to high concentrations of total dissolved solids. So actually in the Permian Basin region, they have a lower salinity compared to the northeastern United States. They can also be enriched in naturally occurring radioactive materials, and they can contain variable concentrations of organic compounds. And spills from oil and gas production, from including hydraulic fracturing fluids, chemicals used to enhance production, and these wastewaters, spills are happening. The expansion and production that we've seen since around 2008 in oil and gas development across the US was also in parallel, we saw an increase in spills and these unintentional or intentional releases of different materials. So the plot here in the upper right is showing the number of spills over time. And New Mexico, where our study is taking place, has seen a little bit slower rise in the number of spills compared to other states like Pennsylvania and North Dakota. But even in the state of Pennsylvania, even if there aren't as many spills occurring, or there are a lot of spills occurring, and they're happening across the entire state. Pennsylvania has actually implemented some new practices that, although the number of spills increased rapidly, new practices have actually decreased the frequency over time.
Distribution of spills to oil and gas well in Pennsylvania line the shale gas basin, parsing from the lower left corner towards the upper right border.
So going back to the, talking about the field site, we're working again in southeastern New Mexico, outside of Carlsbad, New Mexico.
Ites C, D, F, G, H, K, and R were sampled.
And this is an image, an aerial image of the area where the dumps were occurring. Each of these brown lines are oil and gas field access roads, and each of these different squares are actually well pads where production is ongoing. The thing to understand is that the dumps that were occurring, all the ones in green are where we actually sampled, whereas everything in pink is a dump that was identified, but we did not sample during our 2018 sampling campaign. One thing to keep in mind is that although a number of these dumps were occurring on a well pad, the actual operator of that well pad was, their fluids were not what was being released onto the surface, mostly because a lot of those wells were not actually producing wastewaters any longer because the age of the well was such that it was only producing product. So we went out, and in many cases, we had multiple releases on a single well pad. And so an area that was mapped by Terry Gregston shows the extent of how much was released onto the desert landscape from 1 to 3 separate dumps. And so we sampled in triplicate within each of those dump areas. And then we also sampled from a background zone to have control samples to look at what the geochemistry and microbiology of the environment was prior to dumping.
An overhead view of a site including a square space connected to roads with a background sample area circled off to the side of the site, and a red and yellow dumping area next to the square dump site. Objective: to evaluate environmental impacts due to illegal wastewater dumping, soil geochemistry, soil microbiology, and plant health.
So what we wanted to know is, what are the environmental impacts? How is the soil geochemistry being affected, and how is the whole ecosystem being affected? These dumps could have no effect on the environment, but we don't know that without collecting data. So we use soil microorganisms as a proxy for what the ecological effect might be, and then we correlated that to some of the plant health measurements that our colleagues were able to do.
An image of people wearing gloves putting dry soil into different containers. Methods workflow included 18 dump-affected soil samples, 15 control soil samples, and 4 plant species samples.
So when we went out into the field, we collected surface soil samples from 5 dump sites in triplicate, and then and we included both dump-affected and control soil samples. And we also collected plant samples from a single site, again in triplicate, from dump and control areas. We then brought those samples back to the lab. For the soils, we did geochemistry, where we extracted the soils in water in order to look at the easily extractable anions, cations, specific conductants, and even isotopes. All of this work, again, is done in collaboration with other USGS colleagues and university colleagues. So I just want to highlight how I'm able to work with a really awesome team of other researchers. We also looked at how much moisture was in the soil, the total amount of carbon, total amount of nitrogen, and radium. We then did microbial community analysis. So we're extracting the DNA from our environmental sample. We're amplifying up a gene of interest, which is the 16S rRNA gene. I like to think of that as your identification card or name tag of how my name is.
The gold standard taxonomic gene microbiology.
We're using that magic box to actually sequence those microbial communities.
Characterize community composition, diversity, abundance, and identity of organisms, Mothur R. Filisake, and vegan packages.
And then we're doing bioinformatics to get the answer of who's there. So what is the community composition? How diverse is it? What's the relative abundance of different organisms, and who are they? We also did plant analysis where we sampled 4 separate plant species and digested the biomass to look at cation and strontium isotopic analysis. So as you can imagine, looking at this landscape that has been affected by one of these illegal dumps, there are geochemical changes to the soil from the oil and gas wastewater dumps.
Flat, dry land with a patch of dark brush surrounded by lighter brush.
And so I'm not going to spend too much time on this. I do realize I'm going a little long, but the soil geochemistry is differing between dump and control soils. So I'm just going to highlight here in our specific conductance and percent moisture. For all the plots that you're going to see moving forward, all of our dump affected samples will be in blue and any of our control samples will be in orange. I'm presenting all the data as box plots so that you're seeing kind of the range, but also median in the data.
N equals 18 dump equals 15 control soil samples. Significance evaluated with a Mann-Whitney U-test.
So on the far left, specific conductance, which provides us a little bit of information on like the overall salinity of the soil. You're seeing a much, much higher salinity of these, the dump affected soils compared to the controls. And again, we're in a desert arid habitat. So when we look at the percent moisture, we're going from bone dry soils to having 2 to almost 6% moisture. And so it's really a significant effect on the soil properties from the legal dumping.
Data courtesy of Cozzarelli. Significance evaluated with a Mann-Whitney U-test. Other anions and cetaceans significantly evaluated in dumps. Lithium, boron, calcium, barium, potassium, magnesium, and zinc.
We also did measurement of a lot of trace anions, cations, and metals. And the take home here is that all of a number of elements are really great oil and gas wastewater markers, and they're all significantly elevated. And when I'm talking about significantly elevated, if you look at our far left plot, we have almost no sodium or very, very little sodium present in the soils, but we're looking at having a median of like 3,000 milligrams per kilogram of soil of total sodium in those samples, and going all the way up to 8,000. You'll notice that the boxes are very wide, so there's a much higher range in the chemistry of dump affected soils compared to the controls, which are all very, very similar across the board. And so our data was able to show that the changes we're seeing in the environment, visually are being supported by the geochemistry, so the data we're generating in the lab, and that these effects are indeed due to oil and gas wastewaters and not some other type of material that had been dumped. But all of this comes down to, what is the biological effect
of oil, gas, wastewater dumping on microbes and plants?
Just because we can see a change, we need to know what's happening to the food web, what's happening to this habitat, and how could it potentially affect ecosystem services and potentially trust species. So this picture is actually showing some of the hydrocarbons from one of the dumps that has dried on the soil surface. And each of these little divots here are actually cattle footprints, because these are very important rangelands for the local population that are raising cattle. So when we look at soil microbial community changes
in dump affected soils,
we really want to know, how are those microbes who are the base of this food web being affected? Well, they are being affected. Another evidence of how this is really important habitat these are actually bobcat footprints on the landscape. And so we're seeing, these visual changes, but really what we want to know is, how can microbes be used as a proxy to understand what type of higher trophic level effects there might be? So since I'm going short in time, I'm going to skip over a couple of the slides and really focus on some of the highlights, or go quickly through them. First thing that I'll show you here is that we're seeing lower overall microbial diversity in our dump affected soils. And so that's an important piece of knowing that, if the who's there is being changed, what else might be happening? So we have colleagues that have done some follow-up studies to even look at plant diversity and how the vegetation might be affected, whether or not we're going to see lower diversity over time due to these effects. And then we also wanted to know, are the changes, are these lower diversity really associated with changes due to the geochemistry? And so this is one of those multivariate plots, and it really shows us that the different geochemical parameters that are associated with oil and gas wastewaters are driving the shift in microbial communities.
Non-metric multidimensional scaling NMDS, ordination of a weighted unifrac distance matrix with significant geochemical vectors fitted using NFID.
I'm not going to spend a lot of time there because I want to talk about who changed, right? We can see the changes, but who changed and why is it important? So this plot is showing all the different, the dominant taxonomic groups that we found in our soil samples. On the left are our control microorganisms found in our control soils, and on the right are dump. And the circles that are largest in our dump samples compared to our control include the members of the gamma proteobacteria class and the Firmicutes phylum. And so these organisms potentially could have very different function compared to kind of your background standard desert soil taxa, who are often in higher relative abundance, like the actinobacteria, the acidobacteria, or the verrucomicrobia. But when we drill down a little bit and look to see, all right, who's really increased in abundance in our gamma proteobacteria, we found that these 5 families of bacteria who are in higher relative abundance in our dump affected soils include taxa that could be really important to remediation or attenuation of the dumped contaminants on the landscape. So we see that halophiles or salt-loving microbes are much higher abundance. Native desert soil bacteria are not going to be able to tolerate high concentrations of salt because they're not ever exposed to it. But we're seeing that these microorganisms that can tolerate up to 25% sodium chloride are being selected for in this environment. We're also seeing anaerobes. Typically, these soils would be only aerobic. And so the fact that we're now seeing a shift towards anaerobic taxa is important. And that also includes taxa that are known to be able to degrade hydrocarbons. There are also organisms that are present in oil and gas wastewaters and other hydrocarbon contaminated environments. And this is important because there are a lot of strategies out there for remediating hydrocarbon spills in freshwater systems. But very rarely, to my knowledge and talking with collaborators and other stakeholders, there's no good way to remediate, no effective strategy at the ready to be able to deal with hydrocarbons in terms of degrading them microbially, also under high salt concentrations. So this is a very good way of thinking about how can we develop new remediation strategies that can be important for restoring the environment.
Changes in plant health due to oil and gas wastewater dumping. An image of broom snakeweed shows a control that is more green than the dump example.
So we wanted to know whether or not these changes actually were correlating with changes to higher trophic levels. And without going into the data, yes, it does. So we were able to use microbes as a way of getting that base food web analysis to show that the ecosystem is being affected. And then we went and looked at some of the plant health. We see that the dump affected plants are experiencing mortality compared to the control plants. And so this is something we'd like to follow up more on, is how can we develop microbes as being a robust tool for looking at those changes. So I'm just going to go ahead and jump through to here and talk about our key findings. So from that project, we're seeing that wastewater dumps are changing geochemistry and the microbial communities, and all this is associated with signatures of oil and gas wastewaters. And the elevated salts and we're seeing in the soils are also being reflected in the affected plants, and all of that's coming together of being able to see the effect from the abiotic system of the soils itself through the microbes all the way up to the plants. And it gives us a proxy for or gives us some knowledge that additional ecological studies are going to be needed to see how all the different trophic levels might be affected. All right, so in order to kind of wrap up here, how can we use microbial heroes? Well, they can be bioindicators of exposures. Microbes can serve as fingerprints of a spill, and they can indicate food web shifts and changes in biogeochemical cycling. Why is this important? Well, to grab a microbial sample, all you need is a sterile container and a freezer. So we can collect soil samples and maybe 50 grams of a soil sample at a site, put it into a sterile container and freeze it. And that's a lot more straightforward in terms of a sampling campaign than trying to do a full vegetation survey or trying to collect higher organisms. So if we can use microbes as an indicator of food web shifts, it could potentially help to prioritize where additional food web types and higher trophic level studies are needed, and also be even a way of understanding effects without having to do more long-term studies. We also see that there's potential for biodegradation of contaminants. Native microbes are being selected for by spills, and they could be really good targets for doing in situ bioremediation or natural attenuation. And so, just to highlight an example that if we're getting salt-loving microbes that can also degrade hydrocarbons, maybe we just need to figure out how to make them really happy in the Permian Basin sites in order to promote restoration, the restoration trajectory for those sites. When it comes to recovery and restoration, we really need to be thinking about how the restoration trajectory can support the return of microbial communities to baseline. How can we make sure that things like nitrogen cycling, phosphorus cycling, decomposition, and photosynthesis by microorganisms, how do we get those processes back so we can have a really healthy, stable environment, ecosystem, excuse me. And, kind of thinking back to bioindicators of exposure, it's possible that microbes could even be an indicator of a species, of an ecosystem returning to baseline and of a successful restoration. Because if we see those processes coming back, then we can know that we're on the road to recovery and maybe have a potentially stable new environment post-restoration. So with that, I'd like to acknowledge all of my many collaborators and our funding from the USGS Environmental Health Programs and the Energy Resources Program.
Acknowledgements. Logos include UIEP, USGS Science for a Changing World, Geology, Energy and Mineral Science Center, Reston Microbiology Lab, and the National System of Public Lands U.S. Department of the Interior Bureau of Land Management. Thank you to Isabel Cozzarelli, Jean Gish Robert Andrews, Amy Springfield, Allison Lininger, Yisha Shrestha, Adam Mumford, Terry Gregston, Mark Engel, Doug Kent, Darren Dunlap, Cassandra Harris, Mitra Kashani, Bridget Pollard, and others. Funding from the USGS Environmental Health Programs and the USGS Energy Resources Program.
And with that, I would be happy to take some questions.
Great. Thank you, Denise. Go ahead and raise your hand or drop a question in the chat. To get it going, I really appreciated your examples, one being from the aquatic system and then one being from a more arid terrestrial system to kind of show the breadth of how the microbes might be used. Could you comment, though, on seasonality or temperate versus Arctic systems and the use of microbes?
In terms of things like biodegradation potential?
Yeah, just I noticed we had some of our Alaska friends on the call, and would studies of microbes be approached in the same way in Alaska as they would in Texas, so to speak?
I would say that, yes, I would say the basic methods are going to be similar. The way that you would do experiments would have to shift to look at processes that are more in situ relevant. So whenever I'm developing studies, I'm always taking into consideration the in situ conditions and developing those studies and the methods to really match those. We do know from a number of oil degradation studies, Deepwater Horizon in particular, the rates and pathways are different. And so we do need to be doing studies in different habitats, different geographical areas to know how different the rates will be. So in Alaska, things are going to go slower at certain times of year, but there might also be hotspots in activity depending on the temperature. Temporal shifts, doing a full microbiome assessment of a waterway, for example, even just a pristine one, that's something that we're still moving to as a community. How much variability is in a pristine stream, how temporal that is seasonally, but also with water flow, those are things we don't fully know. And so understanding baseline and understanding return to baseline is still a little bit of our unicorn, and it's something we're moving to. And as a community of microbial ecologists, we understand more and more how important it is. And so those are data we're collecting.
Great, thank you, Denise. Stacy put a question in the chat. Did the Elk River NRDA use your study as evidence of injury?
I can't fully respond to that because I wasn't involved in it directly. So it's not funded by NERDAR. I was involved in the lessons learned workshop that was held by West Virginia University and did interact with some of the folks who were there from like Fish and Wildlife Service who were dealing with the initial response. So whether or not our data were used once they were published, I'm not sure. I do apologize for that, but I'd be happy to follow up on that if that would be of use. Mm-hmm.
Great. There's a couple more questions. I know we're getting close to the top of the hour, but Denise, can you hang on for the last couple questions?
No problem. Sure.
Okay.
I think I'll take Suzanne's question first and then come back to Rochelle. Suzanne, illegal dumping is still going on. And yes, one part of our study is actually to identify those sources. And we're making some progress with that, but I just opted not to show those data since the focus of the results was primarily on the microbial processes. So we have a couple of papers that are in draft form right now. We're including focus on the microbiology, but also another one focused on that source characterization. And that was a big motivation for our study. Mm-hmm. Yeah. All right. So then, Rochelle, I mentioned the microbial communities, their function can be an indicator of return to baseline or pre-contamination state. Were we and our team able to return to Elk River to assess community function a few years out? Unfortunately, we were not able to. That was really due to funding and priority. So since the MCHM chemical is not widely used, so it's not something that has a really large mass, it's not heavily used in coal processing. There's other frothers. We ended up putting our priorities elsewhere for other projects. With the Permian Basin study, though, we do have samples from 2 years later. For the geochemistry, just due to COVID field work, we had to have other folks do some of the field work. So I was not able to be out there myself. We didn't collect any samples for microbial community analysis, but we did additional vegetation sampling and sampled for geochemistry of those soils. So we're hoping that that might be able to provide more of an indication of that baseline, at least from the geochemistry, and hopefully that will drive more future work of trying to look at the microbial baseline. Thank you, Sarah. And great. Did I miss any other questions?
No, you just had a comment there from Sarah about the similarities between plant communities. I did drop in Denise's email if you'd like to follow up with her on any questions. But Denise, thanks again for your time today and a really interesting talk. And we look forward to seeing everyone next time. Have a great rest of your day.
Thank you.